Thank you very much for this opportunity to present. My name is Jeewan Pandey, and I'm from Texas A&M Potato Breeding Program. So today I'm going to present about the genetic basis of stem and chip defects and strategies for implementation of genomic selection in potatoes. So we have Doug and Jeff. Doug kind of do the chipping, and then he also does the grading, and Jeff maintains the data for the program, like the historical data. And we have Isabel Valles, who is the program director. And yesterday, Marcelo mentioned about the tools for Polyploid Plus, and she is leading the potato team. So we are representing the Texas program here, and we have the poster for Amaga. And you can visit the poster during the poster sessions. So let's introduce the famous crop potato. So it's a very important crop in the US as well as worldwide. Potato chips are the most consumed snack. So most of the potatoes goes for processing. Out of that, some are French fries and most of them are chips. And for consumers, the appearance or the aesthetic of the chip is very important. The consumer demands uniform and light colored chips. And also it's important for the producers because the processor uh, adjusts the price of the potato based on the chip quality. And there are some undesirable traits in potato, which includes the internal heat necrosis, the vascular browning, hollow heart, brown center, dark colored chips, and stem end browning. So my talk today just focus on the stem end browning, which is also called stem end chip defect. So I have kept the potato picture over there. So it has two ends. One is the apical end, and the other one is the stem end. The end that is connected to the mother plant through the stolon is called the stem end. And if we fry the potato chips, then we see the color differences between the apical end and the stem end. Sometimes on the stem end, we see the browning. That's called the stem end chip defect. And it has cost around $10 million some years to the potato industry. And from Wisconsin, Jeewan and Paul Bethke has done a lot of research on stem and chip defects, and they have developed this severity score ranging from 0 to 5, 0 being none and 5 being mostly affected or severe. And there are different studies identifying what are the causes behind the stem and chip defect. And a lot of them have reported about different stressful growing seasons. And the one mostly reported is the heat stress. And some greenhouse studies reported by Paul Bethke shows that the transient heat stress for 7 days is enough to induce the stem and chip defect. And in Texas condition, under normal field conditions, we have high temperature. You can see on the growing season, there is more than 25 degrees Celsius for several days. And we see a lot of stem and chip defects under field conditions. And the bad thing is the cultural practices and chemical treatments has not been effective. But good thing is there are some varieties which shows the differences. And I think it's the best strategy to improve the chip quality. So our objective is to evaluate the germplasm under field conditions and investigate the genetic basis of the stem and chip defects, and also develop some genomic selection strategies to efficiently select the against the stem and chip defects. So for this study, I used some of the national chip processing trial data as well as the regional chipping trial data. So out of 841 total clones phenotype for several years, I had genotypes for 384 clones. And we had Atlantic, Snowden, and Lamoga as the reference varieties. For the stem and chip defect traits, I use 3 different traits: incidence, severity, and the index. So incidence is just the presence of the stem and chip defects. So our trial was different. So we sometimes had 10 chips, sometimes we had 20 chips, so I adjusted based on the percentage. And also we had the 0 to 5 scale. And for the severity as defined by Paul Bethke, we had a 0 to 5 scale. And for the index, I multiplied incidence with the severity. And for the GWAS analysis, I use the GWAS poly, and for the prediction, I use the stage-wise. And when we have the phenotype, it's always good to observe the outliers and check the normality of the residuals. And this is one of the methods that Dr. Engelman has presented in the stage-wise, just to observe through the QQ plots. The straight line indicates the normality, and the points that are deviating from the straight line are the possible outliers. So for the breeding programs, it's very obvious to see some of the outliers, but it's worth to check for the outliers. And what I have understood at this point is not all of the outliers are the wrong data, but it should be inspected quite well. And regarding some of the summary statistics and the heritability, based on the percentage of incidence, we have 24.78 as the blues percentage for the incidence, and it ranges from 0 to 100. We had some clones, like out of all chips, they were having all the stem and chip defects. And the severity blues is 1.34, and for the index is 2.41. And all these three traits are lowly heritable. We can see the heritability ranges from 0.12 to 0.17. And if we dive into the GWAS analysis, we found some QTLs in different chromosomes, like 5, 6, and 12, mainly with the dominant model. For the incidence, we got QTL in 5, 6 chromosomes. And the percentage of phenotypic variance explained is around 4% to 9%. And let me introduce some of the factors which determines the prediction accuracy. So it is connected with the trait architecture, some of the marker properties, some of the training population, and there are different genomic selection models applied. But today I'm just going to use the marker density. We have 4,000, 10,000, and 15,000 markers that were obtained through different versions of the SNP array. So I'm just using those 3 sets of SNP markers to see the prediction over those 3 stem and chip defects. And for this one, I use the Pearson correlation coefficient for the expected and the observed values, and integrated the 5-fold cross-validation with the stage-wise. Yeah. I use the mask function to mask some of the phenotypes, and I use that feature to do the cross-validation. So I distributed all the data randomly into 5-fold, and in each fold, the remaining 4-fold data is used to train the population, and the remaining 1 set is used as the testing population. And it went all to the 5 round, and I repeated for 10 times. And regarding the distribution of the SNP panel, we can see from the heat maps that all of the SNPs are distributed in all chromosomes. So at least on the lowest 4,000 panel, we even had 216 SNPs on chromosome 10. So that was the lowest number of SNPs we have. But we have covered all the chromosome. And that was basically designed initially with the small array. Also, it covered all the genomes. And it's not surprising that the centromeric regions has less SNPs than the ends. And when I analyze the data for different traits based on the different set of markers, we got the prediction accuracy. There is, you can see how much it increased. But in terms of cost efficiency, since we want to use less number of markers, So I'm trying to present in that way there. If using less number of markers like the 4,000 instead of 15,000, then the prediction accuracy decreases by 6% for the incidence, 3% for the severity, and 2% for the index. And I'm planning to expand this study further, but in summary, I think the Texas field trial can work as the heat screening site for some of these useful traits. And Amaka is presenting some of the work related with the heat screening, and she has a poster. But we identified some genomic reasons, and it will be worth to look over those reasons and find some genes underlying those mechanisms, because there are some genes that differentially express between the apical region and the stem end region. Maybe those should be connected in a way. And my conclusion for the marker density is some of these moderate marker density is useful enough to predict some of the incidence and severity and index. And as Dr. Endelman presented, I'm gonna use some of those imputational features newly released, because we have some set of markers that only has few thousand markers, but we can impute now to higher density markers. Not only that, we can also use this model to expand some of the factors that affect the genomic accuracy. I am planning also use the alpha CMR in the future as well. And with that, I'd like to thank all the team members of the potato breeding team at Texas, Rose Lab, all the collaborators of the NCPT trials, and we have Spring Lake Potatoes and CSS Farm. And thanks for the tools for Polyploid for putting this together.